I have a couple of questions that are posted already in the in the Google form that was shared. But I'm not sure how many more will be joining this program. So let's see if you have any queries and questions. You may post it using the chat box as well next to the YouTube. So we will take up each of the questions as it comes. And this is the last of the two discussion sessions that we have. And we are going to discuss the questions. Okay. Okay, the first question is that gene sequence of cancer cells and associated immunotherapy by Neha Acharya. I'm not too sure about the question as to what exactly Dr. Acharya, I guess, wanted to sort of discuss, but I assume whether gene sequencing would identify any of the markers and so on. So this is a possibility. But what I wish to say is that the gene sequencing normally is not used in this kind of a thing, that you can use any of the cell for the sequencing. But if you're talking about expression levels and so on, then people do RNA sequencing and compare. For example, this the tumor cells as to how they express differently as compared to the normal cells or the cells surrounding the tumor or cancer. Or after, for example, therapy, whether is there any signature that is different as compared to the one that was there before the treatment began. So this kind of an analysis would help one to identify markers that could be used as a readout as to how unique or how efficient that drug or therapeutic approach that has been employed. Since the question is not that clear, I'm not too sure about what is being exactly asked. If Neha is around, you can give a more elaborate questions, and we'll be able to answer that. Then there is another question by Shiv Om Pratap on progression of genomics. Again, I'm not too sure what exactly is being asked, but if it is about how the field genomics evolved, then it is already covered in the topic, and we also have given you the link for reading more in terms of reading material. But essentially, what the lecture spoke about is that from say 1990 to early 2000, that's when the genome or functional genomics or genomics, this particular field, sort of exploded. What are the major changes took place? That is with the announcement of the the genome sequencing human genome project in 1990, and then the chromosomes are mapped, and then many genomic fragments were cloned in library and so on. Few of the chromosome specific sequencings have been released, and then people took up other models, for example, mouse, yeast, and others that they're able to complete. And then came, multiple sequence models that they have been, like whether it's Arabidopsis, whether it's Drosophila, whether it's C. elegans, one after the other. The genome sequence was sort of completed, and in 2001, human genome sequencing, and then and the mouse genome. So that is how this whole thing evolved. Basically, what we have covered is that how the field of genomics, has become from a small lab-based research that what you define as genetics by a few individuals sitting with the microscope and few of the model systems. They were able to convert that into industry scale where the labs have become like industry. You have equipments that are high-end equipments and the robotics, even for example, transferring the solutions from one tube to the other. Earlier, people use pipettes, and then we have the robotic machineries come. Then you can program it, and it will go on adding solutions and so on. So the entire process of sample processing and analysis is sort of scaled up in a way that is done in an industry, and as a result, you have a huge amount of data that is coming in, and that was very difficult to analyze. That's why even computing powers have been brought into this field. So we have a new field called bioinformatics, wherein you have a lot of genome data, we have a lot of variation data and so on that is being analyzed, and certain patterns were identified, and later people started testing whether a given pattern or signature really mean what you see in certain phenotype and so on. So that has sort of exploded because of the fusion of the approaches used to understand the genome as to how it functions with the technologies that have developed. Even now we talk about we have sequencing machines which can really sequence genome at a pace that was simply one couldn't sort of even expect that it can happen when the genome sequencing began. It took ages to complete human genome project, but now it can be done in a couple of days. So that's what I would define as that. But this is very well sort of explained, and then there are links that are given. I'm sure you'll be able to read more, and you can get more information. So there was a question by Dinesh Raj that is there any industry or university working in personalized medicine in India? Right now, no, but certainly there are certain genetic disorders wherein you have defect in a gene. As a result, you have deficiency in the protein that gene codes for. And people go for what is called as therapy, wherein that, whatever the protein that is defective that can be given in a different ways. And again, that's something like is something like a personalized medicine, but still the very concept of personalized medicine has not evolved to that extent, because we are not understood all the signatures that define whether you would respond well to the drug, or how you would respond to a therapeutic regime and so on. So it is still a nascent field. Right now, I will not be able to tell about industry, but yes, in lab scale and some medical schools, there are some programs that have been started. Yeah. If there are any other questions, you can post them. We'll be able to answer. In current trend, how pharma industries are using functional genomics. There are number of approaches people take, and the pharma, when you talk about the classic way of pharma where they use the medicinal chemistry, they don't really use functional genomics, but the more modern biotechnology based pharma companies, they certainly, using that. Even whatever we know now, even when the COVID outbreak took place, people are using that as an, opportunity to understand how we can compare the genome sequence with how the, say, virus respond to a kind of a drug and so on, right? And even, for example, now they talk about the vaccine. There are different, vaccines. Some are RNA-based vaccines. Some are, the adeno-associated virus-based vaccines. And then we also have now DNA vaccine coming up. Now, how good they are in terms of their response to a new variant is again a kind of a functional genomics approach, right? So you have COVID patients who have certain symptoms, moderate, severe, or acute symptoms, and then whether they have been vaccinated, not vaccinated, whether what kind of vaccine is given, and how they respond. This itself is a kind of a functional genomic approach, because they're not only looking at the host, that is the person who is infected, but we are also analyzing the genome of the agent that caused the infection, in that case, say, the COVID virus. Therefore, that you sequence the genome and look at what kind of variation is there. So all that has been delta variant and so on. That particular variant becomes far more virulent, and then you try to compare and then say which vaccine or which drug can be more effective. Is indeed a functional genomic approach, because during the COVID time, people could not do the kind of clinical trials normally people would do for many other disease. Here, everything was done in war footing because really don't have time left, because you are still in the midst of the infection. So that's is a real time example as to how powerful these approaches in terms of validating the efficacy of whatever therapy that you bring in. And so I think that answers your question. Yeah, if there are any other question, you may want to post it. Therefore, we can take it up for discussion. I guess there are no other questions, so we will end the session. And in case if you have any other question, you may continue to post them on the course portal. We'll be able to answer. And all the very best for your exams, and best of luck.